This is Brain Ponderings with Mark Mattson, conversations with scientists at the forefront of brain research. Today, Avi Nath is my guest. He's a friend, longtime friend, collaborator, and currently he's the clinical director at the National Institute of Neurological Disorders and Stroke, where he's also chief of the section on nervous system infections. He was born in Canada and raised there, I think, in Saskatchewan, and then got his medical degree in India, in the Punjab. Then he came back and went to Texas and got his residency at Houston, University of Texas Health Medical Center in Houston. Then he went back to Canada for a few years on the faculty at University of Manitoba. Then he went to University of Kentucky, where I met him, and we started some collaborations, which he may talk about. And then after I moved to the NIA in Baltimore, within a couple of years, he moved to Baltimore and was a professor of neurology at Hopkins. And then about a little over 10 years ago, he was recruited to be the clinical director at NINDS. His expertise is in neurovirology, infections of the nervous system, and that's what he's going to talk about today. So Avi, welcome, and can you just tell our viewers and listeners, what is a virus? What defines a virus?
First of all, let me just say that it's such a pleasure to see you, Mark, and thanks very much for having me on the podcast. I remember our years together with great fondness. And we did a lot of things with HIV together and some very critical discoveries. So they still, to this date, are some of the seminal pieces of work. So I'm very grateful for this collaboration. So the virus, it used to be that most people didn't know what a virus was. Now, even a 5-year-old will tell you, yes, virus. Everybody knows coronaviruses, and they all think it's probably red and white in color. But in fact, viruses have been around as the longest living forms on this planet. So they're the simplest of all organisms. And they've been here long before humans ever got on this planet. And they have a little piece of either RNA or DNA in the middle, and they have some proteins around them. And so those proteins protect the RNA or DNA in there, which is really the genetic sequence of the virus. And that allows the virus to then move around in that protected armor that it has.
And so the viruses, they can't survive and replicate by themselves. They have to get inside cells. Is that right? And
yeah.
And how do they get in the cell, and then what happens when they get in the cell?
So because they're so small and it's a very simple organism, it has to multiply. And so it doesn't have all the machinery that's available to multiply within the virus itself. So what it has to do is it has to get into some other cell, and there then it uses the machinery of that cell to then multiply. So as I said, they largely you can divide them into two types of viruses, with the RNA virus or the DNA virus. But what they do is they just latch onto the surface of the cell, and they bind to what is called receptors. So it's like a lock and key kind of mechanism. Here's the receptor, here's the virus, it kind of locks in there. But then they fuse together, the membranes fuse. And once they fuse, then what it can do is it can just unload all this genetic material inside the cell. And because now the proteins are not really necessary. So once the genetic material gets in there, then the cell has its own DNA and RNA and always making proteins. And now the cell cannot tell the difference between the virus and its own RNA or DNA. So the virus fools the cell and it starts multiplying. Now it can do one of two things to the cell. It can multiply to such large degree whereby the cell will actually die. The cell cannot even handle that many viruses. Or it can form very small amounts, and then it can just hide in that cell forever, and from time to time just release small amounts of its progeny. And they go on. So that's called a persistent infection.
And how, so the genetic material in the viruses codes for the proteins that form that surround the RNA and DNA, the viral proteins.
Yes.
And how, so the virus hijacks the cell's genetic machinery.
Yes.
The cell produces those proteins, and then how do those proteins and RNA or DNA reassemble into a new virus?
Yeah, so the proteins have RNA or DNA recognition domains there. So there's something called the capsid, and the capsid is where the RNA or the DNA binds to it. And there's a tail end of the RNA or DNA has a little loop or something, it's kind of a structure. That structure is recognized very precisely by that capsid. So it won't just take any RNA and encapsulate it, but it'll find just the viral RNA out of the milieu of other RNAs in the cell, and they interact, and then the capsid just kind of binds it close, right? So once that's bound in the capsid, for certain viruses like the adeno-associated virus, then that's all you need is the capsid, and it will release. But there are others that are called envelope viruses. So then the capsid holds on to it, but then the capsid has another domain where there's the envelope comes on top of it, and it encases the whole thing together, right? And so that's called an envelope virus. So for those viruses that have an envelope, they have to go through the receptor mechanism. The ones that are just capsid, they go through what is called the endosomal mechanism. They can penetrate the cell membrane a lot easier through the endosomal mechanism.
Okay, so things like HIV and COVID, SARS-CoV2, they have the envelope protein.
Yeah, they have the envelope. Yeah, for the COVID-2, they call the spike protein. So on top. But
now there are many viruses that infect the brain and other parts of the nervous system, for that matter. Before we focus on HIV and SARS-CoV-2, could you talk about some? I mean, there's a very interesting. Everybody's probably heard of rabies.
Oh yeah.
But there's, I guess I don't know, but probably hundreds of viruses that are known to and can get into the brain or nervous system.
Yeah. So that's is very, very true. Viruses love going to the brain because they can hide there for extended periods of time. See, if you are replicating only in lymphocytes, you won't be able to live very long because the lymphocytes turn over so fast, right? A lot of your cells turn over. The cells in the brain, and Mark, you know that better than anybody else, the neurons that are there, they're there till the organism actually dies, right? So, and the turnover of the other cell types is very, very slow. So viruses love going there because they can hide there forever. They can also change the behavior of the person. And so a good example is, for example, you mentioned rabies. So what happens is if an animal gets infected with rabies, its behavior will change and it will start biting people,
Yes.
right? And by that, what happens, it ends up transmitting itself a lot better that way, because coming out in the saliva, and if you change the behavior of that animal and it starts biting people, it has the opportunity to spread. So I don't know if you remember just this last week, and the Capitol building, there was a fox that had rabies.
Oh, yeah.
and it bit a bunch of people in Congress.
Oh, really?
Yeah, a
I,
whole bunch of people got bit by that fox, and the fox had a number of little pups. So they ultimately killed the fox and all the pups, and all those congressional workers who got bit had to get antibody to rabies virus.
yeah, my brother, younger brother, is a veterinarian, and he had to get rabies shots,
Oh, really?
working as a veterinarian.
Yeah, preventively, yeah.
Yeah. Now,
Yeah.
go ahead and talk about the story of HIV and the brain, because it's fascinating. This evolved over decades and decades as treatments came on board and
Yeah.
so on. But I guess start from the beginning. When did you first learn about or get interested in
Yeah.
HIV?
So, I started my residency in neurology in the very early 80s. At that time, the AIDS pandemic had just started in the country, and Houston was, I think, a coastal area, and most of HIV came through onto the coastal cities, because they came through these merchant ships. And so there was a sizable population of it. It was all in the gay population there at that time. And they were developing all these opportunistic infections that nobody had seen before. The cancer doctors had seen them, and because cancer patients will develop opportunistic infections, but nobody else really knew much about them. So they were all getting admitted at the cancer hospital, which is the MD Anderson Hospital, in the bone marrow unit, because these patients were so immune suppressed, they put them in the bone marrow unit. So in the bone marrow unit, you have to wear your gown, gloves, and all that kind of stuff to go and see patients. Neurology usually is a clean specialty. You almost never touch your patients. You just tell them do this, do that. And but as a resident, I had to go in and see them. My attending would just stay outside. So I noticed that these patients had very fascinating neurological complications. And so with the help of one of the attendings there, I wrote up these patients and published them. And so that got a fair bit of attention, and the WHO got very interested in their neurological complications in these patients. And at that time, there was a lot of social stigma against them too. So, people didn't know how it spreads. If you rode in an elevator with somebody who had AIDS, people got really scared. They like take a bath in Clorox or something. It was like. And so, and I had seen several of them. So my department chair said, "Okay, if you like seeing these AIDS patients, you can see all of them." And I was going to all these hospitals and seeing them. And so I published a series of papers while I was a resident. And then later they found out, this is caused by a virus. And I don't know much about viruses, but I got very fascinated. I said, how is this virus causing these massive neurological complications, or can it really? So people didn't believe that the virus could directly cause any neurological complications. They always thought there must be some opportunistic infection that is there. But then there were others who showed convincingly that no, there is virus present in the brain, and these patients can get demented, and the dementia can be very profound. It can occur in a matter of 6 months or so. So I said, okay, it's by viruses. I was young enough to where I said, okay, I want to study the virus, but I don't know how to study them. So they said, okay, if you're interested, and so I contacted some labs that had isolated HIV here at NIH, and they didn't want anything to do with me. So they didn't want a neurologist there. So people told me, well, if you want to study viruses, you should study multiple sclerosis, because surely that's a disease caused by a virus. And so people were hunting for viruses in various diseases. There was really no strong evidence to suggest one way or the other. So in Houston, there was a guy, Jerry Walensky, and he was studying rubella virus. And so I said, fine, I'll study rubella virus in the context of MS. So I learned a lot about virology and immunology, and how to culture viruses, how to do plaque assays, how to make monoclonal antibodies, and cellular immune assays. So I developed a good expertise in virology, immunology through that mechanism. But that has turned out to be absolutely critical because as years have gone by, I've changed a lot of pandemics, the HIV, Ebola, Zika, SARS, a lot of viruses. And so that fundamental knowledge has helped me tremendously being able to adapt it to understand the brain.
Yeah, and then you began to explore actually the mechanism of what's going on. The virus is getting in the brain, so what's it doing to cause damage to the neurons?
So the fascinating observation is that until HIV came around, conventional dogma was that a virus enters a cell, it destroys the cell, and that causes neurological complication. Just as you said, rabies infects neurons, destroys the neurons, and you say, okay, well, sure, you're going to get neurological manifestations just because it destroyed the neurons. Herpes viruses do the same thing. Here, what happened was, for the first time ever, what we found was that the neurons are degenerating like crazy. Okay, you got loss of neuronal loss. But when you look for the virus, there's no virus in the neurons. All the virus was sitting in either these microglia or macrophages and in some astrocytes, right?
And astrocytes are, in addition to neurons, there's cells that they're as abundant as neurons are called astrocytes. And they kind of, they have a lot of important functions in supporting the function of the neurons, even in providing the precursors to neurotransmitters.
Yeah, you have the king, which is the neurons, but then you need all the support staff, you see.
Yeah.
Which are the astrocytes. You can't survive by yourself. And so, yes, the astrocytes, as you said, and you studied it yourself in great degree, they provide all kinds of growth factors. They sop up all the glutamate and all the stuff that comes in, clean it up, take care of potassium that's being released, right? So
and so the HIV infects the astrocytes. Then what's causing the damage to the neurons?
Yeah, well, that's what has remained a mystery for many, many years. But you remember, you and I did some very seminal experiments, and so our hypothesis was that the virus has to be doing something. I mean, the virus doing something to kill these neurons, right? And one of the things that the virus does is, what we figured out is when the virus assembles, people think that all the proteins get incorporated into a virus and they form a viral particle. That's not really true, actually. What really happens is that only a very small percentage of the proteins and the virus are efficient enough to form the viral particle. And in fact, a lot of the viral proteins get released. And so you have defective viral particles and you have free viral proteins that are being released. And you would think that, okay, maybe there's just a inefficient way of the virus packing itself. Actually, that's not true. The virus is very clever. And what it is it's true, it needs a complete viral particle to replicate, but all these other things, proteins that it's producing that are not getting incorporated, can actually cause a lot of other damage. And so it's releasing all these missiles, at the same time. And they're calling it. So that was what I really chased after, because I had realized from virology that viral assembly, when I was studying rubella and something, that the actual viral titers are very few, very small, and there's lots of other protein being produced. So I thought the same thing is probably going to be true of HIV here. And is it possible that these viral proteins themselves might be toxic?
Yeah.
So we went after the TAT protein, because the TAT protein is not required, never gets assembled in the virus. It's really critical for getting the virus to activate the viral genes in order for it to replicate. But then after that, there's no need for it. So what the cell does is, there's no need for it, it throws it out. So I said, if it's being thrown out, let's see if that protein might be toxic. So we were the first people to do experiments with the TAT protein and show that it was toxic. And then some of them we did with you, characterizing the toxicity of these proteins.
And then
Yep.
And in those experiments, we culture neurons.
Yeah.
And we expose them directly to the TAT protein.
That's right.
There's also another protein, GP120, that
The envelope.
has toxic effects.
Yeah.
And I had a postdoc, Norm Howie, who was a graduate student with Jonathan Geiger, right up in.
Yeah.
So this is when you're in Manitoba. This started.
That's right. That's right.
Yeah.
So he worked with Jonathan Geiger, was his primary mentor, and I was his co-mentor. And he's done well. He's now a full professor at Hopkins.
Yeah.
And he worked with you as a postdoc. Right?
So neurons are excitable, and the excitatory neurotransmitter throughout the brain is glutamate, an amino acid. Yeah. And it's well known that neurons can be excited to death if too much glutamate is released, or the receptors are activated too much, particularly under adverse conditions such as reduced energy availability, like in stroke, or in epileptic seizures where the circuits. So I think it was Norm that first showed that TAT, if you take cultured neurons, you expose them to TAT at kind of a level that by itself doesn't really wipe out the neurons. And then you add glutamate at a low level, the TAT makes the neurons more vulnerable to being excited to death by glutamate.
Yes.
Yeah.
Yeah, he showed those synergistic effects, which I think is a very critical experiment. So showing that you can take sub-threshold levels. So I think what Mark, the point that you're making is that you have normal glutamate levels in the brain, right? And that's necessary for its functioning. Yeah. But now if you have TAT and you put small amounts of TAT, by itself, it doesn't do much at that level. But you have a physiological level and a non-toxic level of TAT together, when you have, it actually causes toxicity.
Yeah.
So that's such a clever way for the virus to do it. It can take a physiological level of it and actually make it toxic.
Now, do HIV patients, do they exhibit epileptic seizures more often than?
Yeah, it's not a very prominent symptom, but yes, they can develop seizures. That is true. And
has anyone done EEG recordings, electroencephalogram?
Yeah, lots of data. Yeah.
And there's any evidence of
activity? Yeah.
Yeah. Yeah, because there's a lot of, we think in Alzheimer's disease that an excitotoxic mechanism is occurring. It's very insidious.
Yes.
Occurring over years and years and years, and a few neurons here and there dying. And you mentioned that HIV patients down in, when you're in Houston doing your residency, some of them would exhibit dementia-like symptoms, cognitive impairment. Now, can you talk about then the evolution of finally the discovery and development of drugs for patients with HIV/AIDS, and then how that's allowed them to live, but on the other hand, how it doesn't seem to completely resolve the problems in the brain?
Yeah, so you're absolutely right. I mean, the biggest discovery that we've done really in virology is the antiretroviral drugs. I mean, that's massive effort, and you have to give a lot of people credit as to how that has happened. But they were a game changer, because these patients were dying so rapidly, and now individuals can live close to a normal lifespan. However, the problem with these drugs is that although they prevent replicating virus from forming, they don't prevent all the viral proteins from being formed. So once the thing about it's the reason it's called a retrovirus is it goes from RNA to DNA, and so that's the retro part of it. And because it has an enzyme called reverse transcriptase, And then it has another enzyme called integrase. And what it does is it takes that DNA and integrates it into the chromosomal DNA. So once it's in the chromosomal DNA, now it's there forever. You can't take it out. So these drugs can either prevent the virus from getting integrated or reverse transcribed. But once it's integrated in the DNA, then there are these drugs called protease inhibitors. They can prevent the viral assembly from taking place, but the proteins are still being formed. Now, some proteins that are being formed in the cell will get degraded. They'll go into the lysosomal pathway and they get degraded. But the TAT protein, as we were talking about, it gets extruded from the cell. So it's present in the extracellular space. So it can still cause damage. So I think these patients, just as you are saying, that they still develop neurocognitive problems. It's slower because a lot of the virus has been controlled. But there is a slow decline. And as these patients are getting older and older, they're developing a dementing form of illness, although not at the same rapidity or severity as we'd seen in the pre-antiretroviral era.
Now, do the antiretroviral drugs, the protease inhibitors, do they get into the brain?
They do, but not at very at the same levels as is available elsewhere. But some of the newer ones can get in. They're very, very potent drugs because these are designer drugs. Here the protease was actually crystallized and then they made these drugs. So they act at very, very low levels. So even though the amount that gets into the brain is not very high, it is still good enough to be able to control the replication and prevent the replicating virus from being formed.
Now, we now have really good vaccines for COVID-19, but my understanding is, as yet, there are no vaccines for HIV/AIDS, right?
Yeah.
What's the issue there? I mean, there's nobody, I guess if somebody knew, maybe they would be able to make a vaccine, but
so people haven't given up. So people are still trying to figure out how to do it. And my guess is through the coronavirus, although how terrible it has been, one of the things it's done is taught us new ways of making vaccines. And we've learned a lot about it. So people are going to try to apply that knowledge to HIV all over again. So far, the reason people have not been successful is that the virus, just like the SARS, has been mutating, right?
Yeah.
At a fairly rapid rate. HIV mutates even faster. And even in a single individual, it can mutate, right? So, and you can form lots of different mutant types. So you can make a vaccine against some strains, but the virus just escapes it. So that's why these vaccines fail. Yeah.
Now here's a question I'm not sure about the answer to. So with these for vaccine, the immune response to vaccine, you get lymphocytes mobilized and they produce antibodies, and the brain is thought to be kind of immune privileged. Do you think these vaccines would, well, I guess they would. They'd prevent the initial infection.
I mean, there's something called, I think what you're getting is there's something called a therapeutic vaccine. So some people have come up with the idea that, okay, if you're infected and your immune system is not getting rid of the virus, what if we give you some kind of a vaccine to boost your immune system, and then it will go and get rid of the virus.
Yeah.
Right?
Yeah.
So those are called therapeutic vaccines. So that idea has been around for HIV for a long time. But I think exactly what you were saying is that if you have a therapeutic vaccine, you still won't be able to get rid of the virus from the brain. And if you did cause inflammation in the brain is, a very tight cavity there. It can't sustain much inflammation. So let's say you did get the cells in there to go and kill all the infected cells. You would cause inflammation in the brain. You might end up actually causing more damage.
Yeah. Yeah.
Right? So
before we move on to COVID, now initially we call it like HIV dementia, right? And then I guess, was it NIH that came up with this HIV-associated neurocognitive disorders? How common is this in patients with?
Yeah, so the terminology is kind of confusing. So Dick Price called it AIDS dementia complex. And then the group in San Diego came up with, they said, no, we call it HIV dementia. And then they didn't quite agree. And then finally there was a consortium put together, and NIMH helped put together these people in Frascati. And and there the consortium then decided to use the term HIV-associated neurocognitive disorders, or HAND. But it's a mixed bag, you know. So you do have the HIV-mediated stuff, but then the current population, a lot of drug abuse is there. There are, other comorbidities that can affect the brains, lipid problems, diabetes, what have you. So they all get sandwiched into this HIV-associated neurocognitive disorders. And now there's a new push to see if we should look at it more closely and try to subdivide it into different aspects. Not all of it is all the same.
Now, risk factors with COVID, the risk factors for poor outcomes and death are obesity, type 2 diabetes, asthma, immune compromised immune function. In the HIV patients with dementia, are patients who say are overweight or have type 2 diabetes more likely?
Yeah.
Yeah.
You're absolutely right. All these lifestyle things do make a huge difference to HIV-associated cognitive problems too.
So they're probably making the brain more vulnerable to the virus and these viral proteins.
Yeah, you're absolutely right.
Okay, and in fact, we think now that obesity and type 2 diabetes is a risk factor for Alzheimer's disease.
Yeah, that makes sense.
Okay. You've been doing it. So lately you kind of, I guess, put HIV on the back burner. And you've been working on SARS-CoV2 and I think some of the first work actually looking at the brains of people who had died.
Yeah.
Can you talk about that?
Yeah, so the thing is, but with each pandemic, what happens is that people don't think about the brain. They get really focused on the primary organ that is affected by the virus. So for example, with AIDS, it was the same thing. People got immune deficiency. Everything was just, on the lymphocyte there. And the same thing with Ebola. It was like everybody was focused on the GI tract over there. And I was one of the first people to go to Liberia and identify these patients were actually dying of meningoencephalitis, at least 50% of them, not of GI illnesses. So when SARS-CoV-2 came around, and in China, it was being recognized that yes, some patients are developing some neurological symptoms. I caught on immediately. I said, listen, this is going to be the biggest problem over here. People are all focused on the lung. Nobody's thinking about the brain. So I said, the only way to figure that out is I need to get some brain tissue, autopsy tissue. Now, getting autopsy tissue was no easy task because nobody was doing autopsies on these patients for fear of getting infected. And in order to take out the brain, it's even more problematic. You have to cut through the skull. And when you cut through the skull, you produce this sawdust that goes everywhere. Oh. And so you have to contain it somehow. You need what is called a vacuum saw. Vacuum saws were not available at that time. They were out of supply. And then you need a level 3 facility whereby all the air can be handled as what's called a negative pressure room. So all the air gets sucked and it has to have HEPA filters and all that kind of stuff. You need PPE. So people were not doing that because they didn't have, either they didn't have the saw or they didn't have a level 3 facility to do the autopsy, or they didn't have PPE. The people in Yale, they had the other two, they didn't have PPE at that time to do the autopsies. So I called around everywhere and see if anybody was doing autopsies. I finally got it from two places. One was the New York Medical Examiner's Office. And there people were dying, in droves and coming to the medical examiner's office there. And when I called them, they said that, yeah, we have 200 bodies here sitting in refrigerated trucks. Okay. And they said that, well, if anyone is COVID positive, we don't even touch them. They just go and either they dispose of the body, however they were doing it. But what they didn't realize is that they were doing all these autopsies where they did not know that they were COVID infected early in the pandemic. So they actually had COVID autopsies that they've done, right? But retrospectively, they knew, these guys were positive. So through a circuitous route, I managed to get 40 brains from. And and then I got another.
And for that, do you have to get permission from the relatives, or how? Or you don't? How does that work?
So yeah, so this is the interesting thing is that once you die, the Constitution no longer protects you. You're no longer a citizen.
Okay.
You have no rights. So the best thing to do is don't die.
Okay. But yeah, I get it now. So these are people who, would not have been buried like at a funeral.
Yeah, so they were interesting. Anyway, so what happened, the reason they end up at the medical examiner's office, they die under very difficult circumstances, right? So these patients, there were a number of people who just died on the subway. They were found dead on the subway.
Oh my God.
Or they're dead on the street. There were some people who died in their sleep at home. So they become medical legal cases. And so they end up in the medical examiner's office. Then they have to decide if they think there could be foul play or whatever it is, then they have to do the autopsies.
Okay, so you got these brains from New York, and now you've got them back at Bethesda in your lab, and you look at them under the microscope. How did you examine them, and what did you find?
Okay, so what we didn't know where to look, right? Where are we going to find where to look? So I said, what we should do is, why don't we do an MRI scan of the brain? And just like we do of live people, I said we could do it of the autopsy brain. It was all fixed in formalin. We got all formalin fixed tissues.
Yeah.
So we have.
And will the formalin kill the virus?
Yeah, formalin will kill the virus.
Yeah. So then it's safe. Yeah.
Safe.
Yeah.
So now what it is that we have high resolution MRI scanners, 11 Tesla, 7 Tesla.
Oh,
but if I wanted to scan anything in those scanners, people will say, oh, no, you're not an MRI person, and we have all these other experiments going on, and you have no access to them. The interesting thing about the pandemic was, at that time, all labs were shut down. Okay, nobody could do anything. These scanners were empty. Nobody was doing any scanner work.
Oh, yeah.
The only research that was being allowed was COVID research. If you were doing COVID, you were allowed to come to their lab. And everybody was trying to figure out, how can I do COVID research? What can I do to do COVID research? So I talked to our MRI guys. I said, oh yeah, sure. Bring the brains, we'll put them on the scanner, and we will take 100 micron sections, and we can scan it all weekend if you want. So usually for an hour of MRI scan is like $500 or $1,000 or whatever it is. It's pretty expensive to do. They didn't really care. Their scanners were empty. Nothing was happening to them. They put the brains and they just scanned them. So we scanned a lot of brains. And we got exquisite anatomy from the human brain that nobody had ever seen before. Excuse me. And then what we started seeing was that there are these blood vessels that look abnormal in the brain. And that the areas around the blood vessels looked abnormal. And they would sometimes see small clots in there. Other times we would see that the blood vessel wall was very abnormal. And sometimes we were seeing what looked like little hemorrhages around the blood vessels. So, but the advantage of the MRI scan, because they're patchy around the brain, they're not everywhere. So now we know exactly where it is on the scan. Now we can take that brain and make a section through the brain exactly in the same area where we see it on the MRI.
Yeah, nice.
See that?
Yeah.
So when we did that, we found that 100% of the brains have pathology in them. So we said, okay, you know what? There must be virus in the brain. Let's do immunostaining and all kinds of the novel techniques, called RNA scope for looking for the RNA. And then we also extracted it and we said, okay, let's do PCR and everything. And we were pretty certain we're going to find a lot of virus. To our surprise, we don't find any virus. There was a lot of pathology, no virus in the brain. So that became a real conundrum for me. I said, listen, I'm a neurovirologist. I need the virus in the brain. There is no virus in the brain. But there's all this pathology, and the pathology is all around the blood vessels. The blood vessels.
And there's not even any, but how are you looking at the virus can you look at it with some antibody under the microscope, or how?
Yeah. So that's what we did. So we took an antibody to the virus. We got several antibodies, like about 10 of them. Whatever people were making was becoming commercially available. We just bought them all. So, because when we initially looked for the antibody will bind to the virus if there's any. And then you put a tag on the antibody and that'll show a different color. So we didn't find any for the first antibody. Then we got the second one. Then people said, "Oh no, you need to use that antibody." I said, "Okay, just give us every single antibody that's available." And we tried them all. Didn't find any. I said, "Well, maybe these antibodies are not good enough. Maybe the RNA. We should look for the RNA."
But do you have a positive control tissue?
Yeah, we made a lot of positive controls. Yeah.
Okay.
So what we did was we took we made these plasmids, which are the synthetically parts of the virus, and not the complete virus, but different parts of the virus. And then we put them into cells and expressed these proteins, and then pelleted them and made a pellet, and then cut them, put them in formalin and cut them and put them on slides, just the same way as we were putting out the tissue. So we had really nice positive controls that way.
But you could have had lung tissue from the same.
Oh, that's the strangest thing. We got some lung tissues. People say that you have all this virus in the lungs. By the time patients die, even in the lung, it's not easy to find the virus. The virus apparently gets cleared pretty fast.
Huh.
Yeah.
Okay. Now the question pops into my mind. You did all this work and we helped a little with TAT.
Yeah.
And then there's this other HIV. Do you think there's a toxic protein produced by the viral genome?
Yeah, I suspect that is true, but we haven't done the experiments yet.
How many? These viruses don't produce many proteins. I mean, in the envelope, for example, I was reading like some of these viruses, maybe 7 proteins, 10. Is it? So
they're not a lot of protein. Yeah.
So it's pretty easy experimentally to.
Yes.
Well, you have to get the proteins, pure proteins, that can be done. Yeah. And then you just see.
Yeah, and then see what happens.
Yeah.
So my guess is that what I would be interested in, because the pathology is on the endothelial cells, I want to see if these proteins will, and Mark, that the endothelial cells in the brain are very unique because they form these very tight junctions, right?
Yeah.
And so in culture, you can actually grow these endothelial cells. You can measure the resistance across them, which is called tear, right? And so what I would like to do is put these proteins on these endothelial cells and see if it disrupts those tight junctions.
Yeah.
Yeah.
Makes
so
it leaky.
Experiment
leaky. Yeah.
Yeah.
Yeah. Huh. Now, what about, you mentioned that the HIV can infect the astrocytes, the glial cells in the brain.
Yeah.
Is there any work going on there infecting, like my son, he's a PhD student at Hopkins, and he'd worked at the FDA a couple of years before he started at Hopkins. And so when COVID hit, he did this collaboration with the FDA people where down there they would infect my son Elliot has these cultured lung epithelial cells.
Oh, yeah.
So they'd infect them with the COVID, and then Elliot would look at essentially effects on gene expression.
Yeah.
And yeah, so is there any work going on like in trying to infect cultured astrocytes?
Yeah, people have done, so you
Or
can,
endothelial cells?
yeah, people have infected endothelial cells. People have infected in vitro, people have taken brain organoids and progenitor cells and stuff and shown that you can infect them in culture.
Ah, okay.
But in vivo, it's been hard to show that that is the case. But in culture, a number of cell types do get infected because the receptor is this ACE2, which is the angiotensin converting enzyme.
Right.
And it's present on
that's what the spike protein binds to.
That's what the spike protein binds.
Yeah.
And so it's present in very large amounts on the endothelial cells. So it kind of makes sense that you can infect them. But and it's present in smaller amounts in other cell types. So in culture, you can do it, but the reality is that the virus itself doesn't get out of the lung very much. Yeah. So if you were to look at the blood of patients and say, can we measure the viral load? Like in HIV, you can measure the virus in the blood, you can measure it in spinal fluid. You don't find circulating virus in the blood or in the spinal fluid of these patients.
Huh.
So it gets stuck in the lung and doesn't come out of there. So now one possibility is that the viral proteins are coming out, but not the virus itself. The other possibility is that you may be forming antibodies to these spike proteins, that the antibodies can then form what is called anti-idiotypic antibodies. And so you have an antibody against the antibody. That idiotypic antibody can look exactly like the spike protein. And then that can go and bind to the ACE2 receptor and cause damage.
Huh.
So our hypothesis is this latter pathway. In fact, we have a paper that just got accepted in Brain 2 days ago. And there we show deposition of immune complexes on these endothelial cells. And that is our hypothesis, that they are idiotypic antibodies to the spike protein that are causing the damage.
Huh.
Viruses have
But there is like is there systemic inflammation with COVID?
A lot of systemic inflammation.
Right. So if in the blood, if you measure levels of what are called these pro-inflammatory cytokines, like tumor necrosis factor.
Yeah.
And then is there neuroinflammation, inflammation in
yes,
the
a lot of inflammation.
A lot of inflammation.
Yeah. So you find these a lot of activated macrophages just sitting in the brain and in various areas. You find more generalized microglial cell activation. So in fact, Michelle Mongi, I don't know if you know her, she's at Stanford. And she studies chemo brain, because in patients who get chemotherapy for cancer, they also develop cognitive problems, and there's a lot of microglial cell activation in the brain. So she asked us to look at these COVID brains and see if we find generalized microglial cell activation. And so yes, we do. We send her that data, and she has a paper currently in revision in Cell, but it's available on bioarchives. And so she claims that the pathology mimics what she calls chemo brain. But the thing is that, yes, there is a fair bit of microglial cell activation. And because of that, you get neuronophagia, which is these activated microglia can then chew up the neurons and cause neuronal damage from that.
You said that in the blood, the patients, even though the lungs are loaded with virus, there's very little virus are detected. But what about viral proteins in the blood?
Yeah, so there's not a lot of published stuff on it, but I've talked to people who claim they can find viral antigens in the blood.
Okay.
And but I'd like to see that in a peer-reviewed publication. I haven't quite seen that quite yet.
Okay.
But my guess is it's still early, and people will eventually publish these things.
Now, I guess let's finish up talking about, I briefly mentioned risk factors for poor outcomes. And I think this is not only true for COVID and AIDS, but I think other infections of the nervous system, that if you're overweight, out of shape, you're more likely to have poor outcomes. Is that true? Like, so you mentioned Ebola. I don't know if there's anything known about outcomes there. And
yeah, but you're absolutely right. I mean, if you look at long COVID, people who are developing these, prolonged symptoms after recovering from COVID, it's very high. Even the death rates of COVID, extremely high in patients who have cardiovascular risk factors or kidney disease, diabetes. They are the ones who are dying at very rapid rates from COVID. So these risk factors are absolutely critical. And because your body is not in shape whereby it can handle an infection. And these infections take a big toll on your body. So if you're not in good shape, you're not going to be able to handle it. And then the mortality rates are extremely high in people who are obese or have high blood pressure and diabetes.
there's one more reason for people, right now to reconsider, if they're not exercising and
Yeah, that's right.
and they're overeating, to reconsider that lifestyle, because we have COVID and there's going to be more viruses coming along.
Yeah, that's the challenge. I used to go to the gym regularly, and then with COVID, I couldn't go there any longer. So I said, now what do I do for exercise? I said, okay, I'll start running. Well, I got problems in my neck. I started getting shooting pains. I said, well, I can't run any longer. So then my daughter presented me a Peloton bike. So now I'm doing it.
Good. That's great. Now there are, I was looking, of course, in preparing for this podcast. I mean, I know a lot about what you've done, but I was reading up a little more on some of the articles you've written recently on. There are viruses that that may lie dormant in the brain.
Yes.
And so viewers and listeners will probably heard of CRISPR.
Yes.
And this is this exciting Nobel Prize winning method for genome editing, essentially, or knocking out the genes. So for example, I guess with DNA virus or RNA virus, in theory, it's possible to develop this CRISPR that will target the virus. Is that true?
Yeah, absolutely right. The thing is, you have to get rid of the virus from every single infected cell to cure it.
Yeah.
Right? So that's the challenge. You may be able to decrease the viral reservoir to some degree, but getting rid of the one last infected cell is very hard, because if you even have one remaining infected cell, it can get activated and then send off its progeny again.
So what other approaches are there to removing?
So your best way of removing it is your immune system. Your immune system can go and recognize all these things. So if there's some way you can manipulate that, that's why CAR T-cell therapy works for tumors.
Yeah.
See that? Because the immune system will go and find these tumors and it'll kill the last remaining cell.
Yeah.
And so now these viruses are very smart. They fool the immune system. They'll suppress them. They'll avoid them. All kinds of things they do. So it's a real battle between the two. But if you can understand how the virus manipulates the immune system, then you can have the ways of being able to block that. So the checkpoint inhibitors is another good example. So you can it will cause exhaustion of the immune. It's viral infections, same thing about cancer, they exhaust the immune system. So you have these cells circulating, but they're no longer doing their job.
Huh.
And that is because it induces the expression of these molecules called checkpoints. Now, if you block those, then they become active again. So it's an interesting concept. I mean, and that the checkpoint inhibitor guys also, they got the Nobel Prize too a couple of years ago.
Yeah. Let's see. One thing I know you got to go pretty soon, but one thing we didn't talk about is that there are hosts, intermediate hosts for a lot of these viruses. In fact, the history of things like Ebola and HIV, where it's very pretty clear, the data, these originally probably came from bats.
Yeah, so a lot of organisms that infect humans come from bats. Okay.
Yeah.
And it's a very interesting relationship between humans and bats. You say, why is it? You know, you get Ebola, there's rabies, there's a whole host of organisms. All the coronaviruses, they come from bats. And so why bats?
Yeah.
So the interesting thing is that bats are cave animals, right? They like to live in caves. And our ancestors lived in caves. In fact, we lived in caves a lot longer than we've lived outside of caves, right?
Yeah.
And so we developed this close relationship between bats and humans, and these organisms were probably going between bats and humans constantly, and they adapted to both species.
Huh.
Yeah.
So, and what's your thoughts on SARS-CoV-2? I mean, it's not clear for sure the origin yet. What are your thoughts on
the origin? Oh, there's absolutely no doubt about it. It came from bats.
Okay.
All of these, there's a lot of relatives of SARS-CoV-2 are all in bats.
Okay.
Right? There are about 8, 9 human viruses that have been isolated, coronaviruses. They all came from bats. And the interesting thing is, each one of them causes neurological complications. All the human coronaviruses do.
But do they cause neurological complications in the bats?
Yeah, that's the thing. So it does cause mortality in the bats. The thing is, the bats can multiply so fast that they can overcome that.
Huh.
Yeah.
Very interesting. So anyway, we should stay away from bats.
Yeah.
Right.
Yeah. Don't go back to the caves any longer.
let them eat the insects.
Yeah.
Right. And let them do their thing, and we just kind of don't bother them.
That's right.
Okay, Avi, it's been great to talk to you. Good to see you. I know you're really busy, and I appreciate you taking the time for this.
Oh, well, thank you for having me on the podcast. I enjoyed it immensely. It was a lot of fun.
Okay, Avi. Take care.
Take care.
Bye.